Good afternoon. So yesterday we have seen what are the different ATP or energy generating metabolites. In that we have seen ATP, we have seen NAD. In the reduced condition, NAD is reduced to NADH, NADH2, and also in plants we have seen the NADP when it is in the oxidized state, and in the reduced state it is NADPH. So today we are looking for the ATP synthesis in prokaryotes. So the objective of catabolic pathway is to synthesize or to make ATP. How ATP is made? ATP is usually synthesized by phosphorylation process or through electron transport phosphorylation process. So when ATP is to transform either chemical energy or in the form of a electromagnetic light energy, usually in photosynthesis or photophosphorylation process, the light energy is converted into chemical energy, and this chemical energy is stored in a high energy bonds of ATP molecule. In simple terms, in photosynthesis process which is taking place in photosynthetic bacteria or in cyanobacteria or in plants, usually light is absorbed and light is getting converted into chemical energy. So chemical energy is the energy form which you observe in the ATP molecule. Now how it is stored? It is kept, or it is usually in the form of a high energy bonds. So cells fundamentally it produces ATP in two ways. One is substrate level phosphorylation process and second one is electron transport phosphorylation. Got this point clear? म्हणजेच काय की प्लांट्स मध्ये किंवा सायनोबॅक्टेरिया मध्ये लाइट एब्सॉर्ब होतो in photosystem 1, photosystem 2, different steps in the chemical in the cascade of electron transport chain. Electrons move from one step to another and ultimately you have a electron acceptor, the last electron donor, etc. But that light energy जी आहे ती कन्वर्ट होते into the chemical form, आणि ती केमिकल फॉर्म जी एनर्जी आहे ती कशात कन्झर्व होते किंवा कशात आपण ठेवतोय त्याला ठेवतोय आपण एटीपी मॉलिक्यूल मध्ये. सो हाउ एटीपी इज प्रोड्यूस्ड? एटीपी इज प्रोड्यूस्ड आयदर बाय सबस्ट्रेट लेवल फॉस्फोरिलेशन और बाय इलेक्ट्रॉन ट्रांसपोर्ट फॉस्फोरिलेशन. सो दिस इज सबस्ट्रेट लेवल फॉस्फोरिलेशन. In substrate level phosphorylation, usually it is the simplest and the least evolved way to make ATP. हा सर्वात सोपा, जुना आणि आधी इव्हॉल्व झालेल्या मायक्रो ऑर्गॅनिझम मध्ये ATP बनवण्याचा हा युजली मेटाबॉलिक पाथवे आहे. So in substrate level phosphorylation, ATP is made during conversion of organic molecule from one form to another. So how ATP is getting formed, or how ATP is made? When you are converting one organic molecule to another organic form, then you can generate the ATP. The energy which is released during the conversion is partially conserved during the synthesis of high energy bond of ATP. So I will explain you with the TCA cycle. So this usually substrate level phosphorylation, it occurs in the TCA cycle, it occurs in the fermentation processes. So in lithotrophic transformations also, these lithotrophic organisms, they utilize inorganic substrates. So among those lithotrophic microorganisms also you can find how to make the ATP. So in Krebs cycle, you know that this is the glycolysis pathway. Okay, so 1,3 glycolytic pathway or the EMP pathway, we have already studied this particular pathway. So I have taken this example. So the 3 examples of substrate level phosphorylation is, that is the 1,3-diphosphoglycerate or glyceric acid. Okay, so when this 1,3-diphosphoglyceric acid is converted to the another form of organic compound, this is the another form, then what happens, one of the phosphate, this is the phosphate from the carbon number 1, it is given out. Okay, and this phosphate is acquired by the ADP, adenosine diphosphate, and when it is acquiring the phosphate group, it becomes ATP, adenosine triphosphate. So when it is getting converted from one organic form to the another organic form, then it is called as generation of ATP by substrate level phosphorylation. Secondly, phosphoenol pyruvate or phosphoenol pyruvic acid. So this enol pyruvate, phosphoenol pyruvate, the phosphate group is added to the second carbon. Okay, so when it is converted to pyruvic acid, then also one phosphate group is released out and is taken up by ADP, adenosine diphosphate. Okay, so when it is taken out, usually, so it is forming the pyruvic acid, and pyruvic acid usually giving when it is converted into pyruvic acid, one phosphate group is taken up by ADP and is converted to, that is ATP. So that is what is called as the substrate level phosphorylation. The third example is acetyl-CoA. This is the example of Krebs cycle. So this is the examples, 2 examples of, that is EMP pathway, Embden-Merov-Parnas pathway. So the third example is the acetyl-CoA. So this is your acetyl group, CH3COOH, acetic acid, and acetyl group is attached to the coenzyme A. Through which linkage? CoSH. Okay. So when it is acting as a coenzyme, then it can also phosphorylate the ADP to make ATP molecule, and acetic acid is given out, and later on the CoA enzyme is also made free and available for another reactions. Okay, so these two, first A and B are the substrate level phosphorylation, and C is the substrate level phosphorylation usually which is occurring in Clostridium and Bifidobacter or Bifidobacterium. And these two are usually observed in many type of microorganisms, means almost all types of microorganisms, mesophilic organisms. So these are the two anaerobic fermentative bacteria which converts acetyl-CoA to acetic acid, and coenzyme is made free, and it does a phosphorylation process in which ADP is phosphorylated to make the ATP molecule. Now we will see the electron transport chain. Okay. So electron transport chain is more complicated metabolic or more complicated method which evolved long after substrate level phosphorylation. So usually the electron transport phosphorylation takes place during respiration and photosynthesis, or during the lithotrophy, or possibly other types of bacterial metabolism. The ATP, or the electron transport phosphorylation, requires electrons removed from the substrate to be dumped in the electron transport chain or the system. So what happens, usually the electrons from the substrates are removed and it is then flown down through step by step in the electron transport system. The electrons usually are transferred through the ATS to some final electron acceptor, as I have said. Either it may be oxygen in aerobic respiration, or it may be a nitrite, nitrate, or hydrogen sulfide, as we have seen in the, that is sulfur bacteria, purple bacteria, and aerobic. In cyanobacteria we have aerobic respiration, that is means the water molecule is the last electron donor and oxygen is generated in the aerobic respiration. Okay, so this is how the electron is being transferred through the system, and therefore you have some electron, last electron, final means last electron acceptor in the membrane. So like oxygen in the aerobic respiration, or you can have nitrate, nitrite, hydrogen sulfide, etc. in the anaerobic processes. So now what happens when there is a flow of electrons, usually there is extrusion of protons. Extrusion of protons means removal of protons from the bacterial or from the prokaryotic cell to the outside. Okay, and because of that, what happens, there is a establishment of proton motive force, that is PMF, across the membrane. So when it is transferring the electrons in this bacterial cell membrane from one level to the another, it is generating a maximum number of protons inside the bacterial cell. So here you will have इसको क्या हो गया रे, just wait a minute, my slide is not moving. Slide is not moving.
करून बघा आता. हँग झालं.
Oof.
5 मिनिटं वेट दे, परत या 5 मिनिटं.
हॅलो नेहा, स्नेहा,
हाँ यस मैम.
स्लाइड पुढे मूव होत नाही आहे, जरा थांबा.
ओके मैम.
Okay. So now we'll see the ATP. The essential component of the membrane for ATP synthesis is the enzyme. How the ATP is made or synthesized? It is the ATPase enzyme which makes the ATP molecule, and this ATPase enzyme is nothing but a membrane bound enzyme which is called as ATP synthetase. The ATPase enzyme, it transports proton and thereby utilizing the PMF or the proton motive force during the synthesis of ATP. So the idea in electron transport phosphorylation is to drive the electrons through the electron transport system in the membrane and to establish the PMF and use the PMF to synthesize the ATP. Okay. So obviously ATP takes a lot more gear than that is substrate level one. Substrate level phosphorylation is very simplest old one, and just when it is converting the one organic molecule to another organic molecule, it can synthesize the ATP or make the ATP. But in electron transport system, it is depending on this particular enzyme, and this particular enzyme, what it does, it transports the proton inside the cell. Means what happens when you are, say for example, you are not able to see the pen now, just okay, let it be. So what I will explain you with the animation later on or tomorrow. So when it is transporting, see, when the protons in the inside of the bacterial cell increases, these protons are expelled out from bacterial cell to the outside environment. Getting my point? Bacterial cell मध्ये जेव्हा इलेक्ट्रॉन ट्रान्सपोर्ट चेन ही कंप्लीट होत राहते तेव्हा आत प्रोटॉन्सची संख्या वाढते. म्हणून काय होतं, प्रोटॉनची संख्या बॅक्टेरियल सेल्सच्या जी आत आहे ती बाहेर पाठवते इनटू द एन्व्हायरमेंट. मग एटीपेस एन्झाईम काय करते, परत त्या बाहेरच्या प्रोटॉन्स ती आत टाकते आणि आत टाकत असताना ती प्रोटॉन मोटिव्ह फोर्सचा उपयोग घेते. दॅट इज व्हॉट इट मीन्स. ओके. सो युजली the example of energy producing and energy consuming functions and use of PMF and the production of ATP is given in the plasma membrane of E. coli cell. So we will see in later in detail about the electron transport system. Now heterobacteria also, or heterotrophic bacteria also, they are the masters of decomposition and biodegradation in the environment, and therefore mainly it brings about two metabolic processes, that is, one is called as the fermentation process and another one is called as the respiration process. So first we will see the fermentation, because fermentation is the ancient mode of metabolism and it has been evolved with the appearance of organic material on the planet. म्हणजे जेव्हा प्लॅनेट्सचं ऑरिजिन झालंय तेव्हापासून त्याचं अपिअरन्स आहे फर्मेंटेशनचं. सो फर्मेंटेशन इज मेटाबॉलिझम इन विच एनर्जी इज डिराइव्ड फ्रॉम द पार्शियल ऑक्सिडेशन ऑफ ऑर्गॅनिक कंपाउंड यूजिंग ऑर्गॅनिक इंटरमीडिएट्स and electron donors and electron acceptors.